What is Mendel's law of segregation?
3, 2, 1, 0.
Each individual has 2 alleles for a gene, which segregate during gamete formation, ensuring each gamete has only one allele. Define linkage.
3, 2, 1, 0.
Linkage is the tendency of genes located close to each other on the same chromosome to be inherited together during meiosis. Explain the term genetic drift.
3, 2, 1, 0.
Genetic drift is the change in allele frequency due to random events, which is more pronounced in small populations and can lead to significant genetic variation over time. What is the Hardy-Weinberg principle?
3, 2, 1, 0.
It states that allele frequencies in a population remain constant from generation to generation, unless influenced by factors like mutation, selection, or migration. Define crossing over.
3, 2, 1, 0.
Crossing over is the exchange of genetic material between homologous chromosomes during prophase I of meiosis, leading to genetic recombination. Describe how sex is determined in humans.
3, 2, 1, 0.
Human sex is determined by the presence of the Y chromosome. XX individuals are female, and XY individuals are male. What is a mutagen?
3, 2, 1, 0.
A mutagen is a physical, chemical, or biological agent that causes mutations by altering DNA structure. What are homologous chromosomes?
3, 2, 1, 0.
Homologous chromosomes are pairs of chromosomes that contain the same genes at the same loci but may carry different alleles. Explain Darwin's theory of natural selection.
3, 2, 1, 0.
Darwin's theory suggests that organisms with advantageous traits are more likely to survive, reproduce, and pass on those traits, leading to evolutionary changes over time. What is genetic recombination?
3, 2, 1, 0.
Genetic recombination is the process by which genetic material is rearranged during sexual reproduction, increasing genetic diversity. Define artificial selection.
3, 2, 1, 0.
Artificial selection is the human-driven process of breeding organisms with desirable traits to enhance or modify species characteristics. What are fossils, and how do they support evolution?
3, 2, 1, 0.
Fossils are the preserved remains of ancient organisms. They provide evidence for evolution by showing changes in species over time. Describe the structure of DNA as proposed by Watson and Crick.
3, 2, 1, 0.
DNA is a double helix with 2 antiparallel strands, made of nucleotide units, a phosphate group, deoxyribose sugar, and nitrogenous bases, adenine, guanine, cytosine, thymine, held by hydrogen bonds. What is the difference between dominant and recessive alleles?
3, 2, 1, 0.
Dominant alleles express their traits even in a heterozygous state, while recessive alleles express only when paired with another recessive allele. Explain the term bottleneck effect.
3, 2, 1, 0.
The bottleneck effect is a genetic drift phenomenon where a population's size is drastically reduced, leading to decreased genetic diversity and changed allele frequencies. What role does RNA play in protein synthesis?
3, 2, 1, 0.
RNA, specifically mRNA, carries genetic information from DNA to ribosomes, where it is translated into proteins. tRNA brings amino acids to ribosomes, and rRNA forms part of the ribosome's structure. What is the central dogma of molecular biology?
3, 2, 1, 0.
The central dogma describes the flow of genetic information from DNA to RNA, transcription, and from RNA to protein, translation. Define natural selection and give an example.
3, 2, 1, 0.
Natural selection is a process where organisms with advantageous traits survive and reproduce. For example, antibiotic resistance in bacteria is an outcome of natural selection. How does genetic variation arise in populations?
3, 2, 1, 0.
Genetic variation arises through mutations, genetic recombination, and sexual reproduction, contributing to evolution and adaptability in populations. What is the difference between homologous and analogous organs?
3, 2, 1, 0.
Homologous organs have a similar structure but may serve different functions due to common ancestry. Analogous organs serve similar functions but have different evolutionary origins. Explain genetic code degeneracy.
3, 2, 1, 0.
Genetic code degeneracy means that most amino acids are encoded by more than one codon, allowing redundancy but protecting against some mutations. What is the founder effect?
3, 2, 1, 0.
The founder effect is a type of genetic drift occurring when a new population is established by a small number of individuals, leading to reduced genetic diversity compared to the original population. Describe incomplete dominance with an example.
3, 2, 1, 0.
In incomplete dominance, the heterozygous phenotype is an intermediate of the two homozygotes, such as pink flowers in snapdragons. Mix of red and white phenotypes. What is codominance?
3, 2, 1, 0.
Codominance occurs when both alleles in a heterozygote are equally expressed without blending, like the AB blood group where both A and B antigens are expressed. Explain the concept of a gene pool.
3, 2, 1, 0.
A gene pool is the total collection of alleles in all individuals within a population, representing its genetic diversity. What is polygenic inheritance?
3, 2, 1, 0.
Polygenic inheritance is when multiple genes contribute to a single trait, such as human skin color, resulting in a range of phenotypes. How does adaptive radiation support evolution?
3, 2, 1, 0.
Adaptive radiation is the evolution of different species from a common ancestor to adapt to various environments, as seen in Darwin's finches. What are pleiotropic genes?
3, 2, 1, 0.
Pleiotropic genes influence multiple traits, such as the gene responsible for sickle cell anemia affecting both red blood cell shape and oxygen transport. Define speciation.
3, 2, 1, 0.
Speciation is the process through which new species arise from existing species due to genetic isolation, environmental factors, or mutations over time. Explain genetic mutations and their types.
3, 2, 1, 0.
Genetic mutations are changes in DNA sequence, which may be point mutations, single nucleotide changes, or frame shift mutations, insertions or deletions. Describe the importance of pedigree analysis in genetics.
3, 2, 1, 0.
Pedigree analysis helps trace the inheritance of traits in families, assisting in predicting genetic disorders' occurrence and understanding inheritance patterns. What is the genetic code, and what are its key characteristics?
3, 2, 1, 0.
The genetic code consists of codons that specify amino acids. It is universal, degenerate, non-overlapping, and nearly always read in triplets. Explain Lamarck's theory of evolution.
3, 2, 1, 0.
Lamarck proposed that organisms evolve by inheriting traits acquired during their lifetime, such as a giraffe's neck elongating due to stretching, a theory now largely disproven. How does the chromosomal theory of inheritance explain gene linkage?
3, 2, 1, 0.
The chromosomal theory states that genes are located on chromosomes, so genes close together are likely to be inherited together due to physical proximity, a concept known as linkage. Define aneuploidy and give an example.
3, 2, 1, 0.
Aneuploidy is the presence of an abnormal number of chromosomes, such as trisomy 21, which leads to Down syndrome. What are vestigial organs, and how do they support the theory of evolution?
3, 2, 1, 0.
Vestigial organs are remnants of structures that served a function in ancestors, such as the human appendix, supporting the idea of gradual evolutionary change. Explain the concept of fitness in the context of natural selection.
3, 2, 1, 0.
In natural selection, fitness refers to an organism's ability to survive, reproduce, and pass its traits to the next generation, influencing evolutionary success. Describe the role of transposable elements in genetic variation.
3, 2, 1, 0.
Transposable elements, or jumping genes, can move within the genome, creating mutations or altering gene expression, contributing to genetic diversity. What is the endosymbiotic theory? The endosymbiotic theory suggests that mitochondria and chloroplasts originated from prokaryotic cells that began living symbiotically within larger cells. Define homologous structures with an example.
3, 2, 1, 0.
Homologous structures are anatomically similar structures due to common ancestry, such as the forelimbs of humans and whales, but may perform different functions. Explain the importance of restriction enzymes in genetic engineering.
3, 2, 1, 0.
Restriction enzymes cut DNA at specific sequences, allowing scientists to isolate genes and insert them into vectors, crucial for gene cloning and recombinant DNA technology. What is a point mutation?
3, 2, 1, 0.
A point mutation is a change in a single nucleotide base in the DNA sequence, which can lead to protein changes or silent mutations, depending on the codon affected. How is mitochondrial DNA inherited?
3, 2, 1, 0.
Mitochondrial DNA is inherited maternally, as it is passed from mothers to offspring through the cytoplasm of the egg cell. What is the significance of genetic counseling?
3, 2, 1, 0.
Genetic counseling provides information on genetic conditions, helping individuals or families understand risks and make informed decisions about health and reproduction. How do homologous chromosomes differ from sister chromatids?
3, 2, 1, 0.
Homologous chromosomes are chromosome pairs with the same genes but different alleles, while sister chromatids are identical copies of a chromosome formed after DNA replication. Define the founder effect with an example.
3, 2, 1, 0.
The founder effect occurs when a few individuals establish a new population, leading to reduced genetic diversity, such as the Amish population with higher polydactyly occurrence. What is gene therapy?
3, 2, 1, 0.
Gene therapy involves inserting, altering, or removing genes within a patient's cells to treat genetic disorders or diseases, such as using vectors to correct genetic mutations. Explain the role of fossils in studying evolution.
3, 2, 1, 0.
Fossils provide evidence of species that lived in the past, showing transitions and changes over time, helping scientists trace evolutionary lineages. What is genetic imprinting?
3, 2, 1, 0.
Genetic imprinting is a phenomenon where certain genes are expressed in a parent of origin-specific manner, meaning some genes are expressed only if inherited from a specific parent. Explain the concept of gene flow.
3, 2, 1, 0.
Gene flow is the transfer of alleles or genes from one population to another, increasing genetic diversity and preventing populations from diverging significantly. Define epistasis with an example.
3, 2, 1, 0.
Epistasis occurs when the expression of one gene is masked or modified by another gene, such as coat color in Labrador retrievers. where one gene controls pigment and another controls color. How does genetic recombination contribute to evolution?
3, 2, 1, 0.
Genetic recombination creates new gene combinations during sexual reproduction, increasing genetic variation within a population and enhancing adaptability. Describe the structure of a nucleosome.
3, 2, 1, 0.
A nucleosome consists of DNA wrapped around histone proteins, forming the basic unit of DNA packaging in eukaryotes, helping to organize and condense DNA in the nucleus. What is adaptive advantage, and how does it relate to natural selection?
3, 2, 1, 0.
An adaptive advantage is a trait that increases an organism's fitness, making it more likely to survive and reproduce, and is thus favored by natural selection over generations. Explain what heterozygous advantage means, with an example.
3, 2, 1, 0.
Heterozygous advantage is when heterozygotes have higher fitness than either homozygote, such as sickle cell carriers being resistant to malaria. Define punctuated equilibrium.
3, 2, 1, 0.
Punctuated equilibrium is the evolutionary theory that species remain stable for long periods, interrupted by brief periods of rapid change. What is gene cloning, and why is it useful?
3, 2, 1, 0.
Gene cloning involves creating copies of a particular gene, which can be used for research, medical treatments, or producing genetically modified organisms. Describe the process of transcription in gene expression.
3, 2, 1, 0.
Transcription is the process by which DNA is transcribed into mRNA, with RNA polymerase reading the DNA template strand to form a complementary RNA sequence. Explain the concept of convergent evolution with an example.
3, 2, 1, 0.
Convergent evolution occurs when unrelated species develop similar adaptations, like the wings of bats and birds, due to similar environmental pressures. How is genetic material transferred during bacterial conjugation?
3, 2, 1, 0.
During bacterial conjugation, genetic material is transferred between bacteria via a pilus, often involving plasmid exchange, increasing genetic variation. What is a genetic load?
3, 2, 1, 0.
Genetic load is the presence of disadvantageous or deleterious alleles in a population, reducing the average fitness compared to an ideal genotype. Describe the significance of outbreeding.
3, 2, 1, 0.
Outbreeding, or mating with genetically distant individuals, increases genetic diversity and reduces the risk of harmful recessive traits. Explain the importance of cytoplasmic inheritance with an example.
3, 2, 1, 0.
Cytoplasmic inheritance refers to the transmission of genetic material from organelles like mitochondria, passed maternally, affecting traits such as mitochondrial disorders. What is stabilizing selection?
3,
Stabilizing selection is a type of natural selection that favors the average phenotype, reducing variation and maintaining population stability. Define the C-value paradox and its significance.
3, 2, 1, 0.
The C-value paradox is the observation that genome size does not correlate with organism complexity, suggesting non-coding DNA plays a role in genome size. What is genetic bottleneck, and how does it affect populations?
3, 2, 1, 0.
A genetic bottleneck occurs when a population undergoes a drastic reduction, leading to decreased genetic diversity and increased susceptibility to genetic drift. Explain the role of RNA splicing in eukaryotes.
3, 2, 1, 0.
RNA splicing removes introns from pre-mRNA, leaving only exons to be expressed in protein synthesis, allowing multiple proteins to be produced from one gene. What is the role of tRNA in translation?
3, 2, 1, 0.
tRNA brings specific amino acids to the ribosome, matching its anticodon to the mRNA codon, facilitating protein synthesis during translation. Define a gene mutation and list its types.
3, 2, 1, 0.
A gene mutation is a change in DNA sequence, including point mutations, insertions, deletions, and frame shift mutations, each potentially affecting protein function. What is directional selection?
3, 2, 1, 0.
Directional selection favors individuals with traits at one end of a phenotypic spectrum, leading to a shift in allele frequencies toward that trait. How do analogous structures support the theory of evolution?
3, 2, 1, 0.
Analogous structures arise in unrelated species due to similar environmental pressures, showing that similar functions can evolve independently, supporting adaptive evolution. Describe the difference between purines and pyrimidines in DNA.
3, 2, 1, 0.
Purines, adenine and guanine, are double ring structures, while pyrimidines, cytosine and thymine, are single ring structures, pairing specifically AT and GC. In DNA, how does the lac operon function in bacteria?
3, 2, 1, 0.
The lac operon regulates lactose metabolism in bacteria, activated by lactose presence and repressed when glucose is available, showcasing gene regulation in prokaryotes. What is allopatric speciation, and how does it occur?
3, 2, 1, 0.
Allopatric speciation occurs when populations are geographically isolated, leading to divergence and the formation of new species due to independent evolution. Explain what sexual selection means with an example.
3, 2, 1, 0.
Sexual selection is a form of natural selection where traits that improve mating success, such as peacock feathers, are favored, enhancing reproductive success. What is a bar body, and why does it form?
3, 2, 1, 0.
A bar body is an inactivated X chromosome in females, ensuring dosage compensation so that only one X chromosome is active, equalizing gene expression with males. Explain the concept of a molecular clock in evolutionary biology.
3, 2, 1, 0.
A molecular clock uses mutation rates in DNA to estimate the timing of evolutionary events, allowing scientists to trace lineage divergence times. Describe genetic drift and its impact on small populations.
3, 2, 1, 0.
Genetic drift is a random change in allele frequencies in small populations, leading to reduced genetic diversity and sometimes fixation or loss of alleles. What are transgenic organisms? And give an example.
3, 2, 1, 0.
Transgenic organisms carry foreign genes inserted artificially, such as Bt cotton, which has a gene for insect resistance. Define mosaicism in genetics.
3, 2, 1, 0.
Mosaicism is a condition where an individual has cells with different genetic compositions, often due to mutations in early development. What is gene therapy, and what types exist?
3, 2, 1, 0.
Gene therapy involves treating genetic disorders by inserting, deleting, or repairing genes. Types include somatic gene therapy and germline gene therapy. Explain the role of ribosomal RNA, rRNA, in protein synthesis.
3, 2, 1, 0.
rRNA forms the core of ribosome structure and catalyzes protein synthesis by facilitating peptide bond formation between amino acids. Define founder effect and explain its significance in evolution.
3, 2, 1, 0.
The founder effect occurs when a few individuals start a new population, leading to limited genetic variation and potential fixation of certain alleles. What is the bottleneck effect, and how does it influence genetic diversity?
3, 2, 1, 0.
The bottleneck effect occurs when a population is drastically reduced, limiting genetic variation and making it more vulnerable to genetic drift. Describe the role of telomerase in DNA replication.
3, 2, 1, 0.
Telomerase extends telomeres at the ends of chromosomes, helping prevent the loss of genetic material during replication in cells like stem and cancer cells. Explain the term allele frequency in a population.
3, 2, 1, 0.
Allele frequency refers to the proportion of a specific allele among all alleles for a gene in a population. Important in studying population genetics and evolution. What is DNA fingerprinting, and how is it used?
3, 2, 1, 0.
DNA fingerprinting analyzes unique DNA sequences for individual identification. Used in forensic science, paternity testing, and studying genetic relationships. Explain the role of biogeography in studying evolution.
3, 2, 1, 0.
Biogeography studies species distribution across geographical areas, providing evidence for evolution through adaptive divergence in isolated populations. What is the function of ligase enzyme in DNA replication and repair?
3, 2, 1, 0.
DNA ligase seals breaks in DNA by forming phosphodiester bonds. Crucial for DNA replication and repair after processes like recombination and Okazaki fragment joining. Describe how linkage and recombination affect genetic variation.
3, 2, 1, 0.
Linkage reduces recombination between closely located genes, while recombination during crossing over increases genetic diversity by creating new allele combinations. What is meant by meiotic drive, and how does it impact inheritance?
3, 2, 1, 0.
Meiotic drive is a process where certain alleles are preferentially transmitted during meiosis, potentially distorting expected genetic ratios. Define homoplasy and provide an example.
3, 2, 1, 0.
Homoplasy refers to similar traits in unrelated species due to convergent or parallel evolution, such as wings in bats and insects. How does genetic hitchhiking occur?
3, 2, 1, 0.
Genetic hitchhiking happens when an allele increases in frequency because it is near a beneficial allele under selection, gaining a selective advantage by proximity. Explain the term transcription factor and its role in gene regulation.
3, 2, 1, 0.
Transcription factors are proteins that bind to specific DNA sequences to regulate gene transcription, controlling which genes are turned on or off in cells. What is artificial selection, and how does it differ from natural selection?
3, 2, 1, 0.
Artificial selection is the process by which humans selectively breed organisms for desired traits, differing from natural selection, where traits are selected by environmental pressures. Explain the significance of Hardy-Weinberg equilibrium in population genetics.
3, 2, 1, 0.
The Hardy-Weinberg equilibrium describes a non-evolving population's genetic makeup, providing a baseline to identify evolutionary changes when allele frequencies shift. Describe genetic recombination in prokaryotes.
3, 2, 1, 0.
Genetic recombination in prokaryotes involves gene exchange through transformation, transduction, or conjugation, increasing genetic diversity in the population. What is a mutagen, and how does it affect DNA?
3, 2, 1, 0.
A mutagen is an agent, such as radiation or chemicals, that induces DNA mutations, potentially leading to changes in protein function or causing genetic disorders. Define sexual dimorphism and provide an example.
3, 2, 1, 0.
Sexual dimorphism is the difference in appearance between males and females of the same species, such as peacocks, males having ornate feathers compared to peahens. How does crossing over contribute to genetic diversity?
3, 2, 1, 0.
Crossing over during meiosis exchanges genetic material between homologous chromosomes, creating new allele combinations in gametes and increasing genetic diversity. Describe the term hybrid vigor, heterosis, and its significance in breeding.
3, 2, 1, 0.
Hybrid vigor, or heterosis, is the increased growth, fertility, and resilience of hybrid offspring, often observed in crossbred plants and animals. What are exon shuffling and its evolutionary significance?
3, 2, 1, 0.
Exon shuffling is the rearrangement of exons within genes or between genes, creating new genes and promoting protein diversity, which can drive evolution. Explain the phenomenon of cytoplasmic male sterility in plants.
3, 2, 1, 0.
Cytoplasmic male sterility is caused by mutations in mitochondrial DNA, resulting in male sterility, used in hybrid seed production for plant breeding. What is a silent mutation?
3, 2, 1, 0.
A silent mutation is a change in DNA sequence that does not alter the amino acid sequence of the protein, often due to the redundancy in the genetic code. Explain phylogenetic tree and its purpose in evolutionary biology.
3, 2, 1, 0.
A phylogenetic tree is a diagram that represents evolutionary relationships among organisms, illustrating their common ancestry and divergence over time. What is the difference between orthologous and paralogous genes?
3, 2, 1, 0.
Orthologous genes diverge after a speciation event, retaining similar functions, while paralogous genes arise from gene duplication and may evolve new functions within a species. Define horizontal gene transfer and give an example.
3, 2, 1, 0.
Horizontal gene transfer is the transfer of genetic material between organisms other than by direct inheritance, such as bacteria acquiring antibiotic resistance genes from other species. What is gene expression and how is it regulated in prokaryotes?
3, 2, 1, 0.
Gene expression in prokaryotes is regulated mainly at the transcriptional level, often by operons, e.g., lack operon, allowing genes to be turned on or off in response to environmental changes. Explain the term genetic recombination in meiosis.
3, 2, 1, 0.
Genetic recombination in meiosis occurs during crossing over in prophase I, leading to the exchange of DNA segments between homologous chromosomes, increasing genetic variation. Define allopolyploidy and give an example.
3, 2, 1, 0.
Allopolyploidy is a condition where an organism has multiple sets of chromosomes from different species, common in plants like wheat. A hybrid are different species genomes. What are retrotransposons and their role in the genome?
3, 2, 1, 0.
Retrotransposons are DNA sequences that can replicate and insert themselves into new genomic locations using reverse transcription. Impacting genome size and gene regulation. Describe how mutations can be beneficial to evolution.
3, 2, 1, 0.
Beneficial mutations provide adaptive advantages, such as resistance to antibiotics in bacteria, which increase an organism's chances of survival and reproduction, fueling evolution. What is the principle of codominance?
3, 2, 1, 0.
In codominance, both alleles in a heterozygous organism are fully expressed, resulting in a phenotype that shows both traits, like AB blood type in humans. Explain the difference between chromosomal and point mutations.
3, 2, 1, 0.
Chromosomal mutations involve large changes in chromosome structure, e.g., deletions, duplications, while point mutations affect single nucleotide pairs, often impacting only one gene. Define phylogeny and its relevance to evolutionary studies.
3, 2, 1, 0.
Phylogeny is the evolutionary history and relationship among species or groups, aiding in the understanding of ancestry and divergence patterns over time. What is inbreeding depression and how does it affect populations?
3, 2, 1, 0.
Inbreeding depression is the reduced fitness of a population due to breeding between close relatives, increasing the likelihood of expressing harmful recessive alleles. Describe the bottleneck effect and its long-term genetic consequences.
3, 2, 1, 0.
The bottleneck effect is a drastic reduction in population size, causing loss of genetic diversity and making populations more susceptible to genetic drift and extinction. What is the role of histone proteins in DNA organization?
3, 2, 1, 0.
Histone proteins help organize DNA into nucleosomes, allowing DNA to coil tightly within chromosomes, and play a role in gene regulation. Explain the concept of a gene pool and its significance in population genetics.
3, 2, 1, 0.
A gene pool is the collection of all alleles in a population, representing genetic diversity and determining evolutionary potential through changes in allele frequency. How does non-disjunction lead to chromosomal abnormalities?
3, 2, 1, 0.
Non-disjunction is the failure of chromosomes to separate during meiosis, resulting in gametes with abnormal chromosome numbers, leading to conditions like Down syndrome. What are genetic markers and their importance in genetics?
3, 2, 1, 0.
Genetic markers are DNA sequences with known locations on chromosomes, used in identifying genetic variation, linkage analysis, and studying inheritance patterns. Describe the significance of fossil evidence in studying evolution.
3, 2, 1, 0.
Fossils provide direct evidence of past life forms, documenting evolutionary changes over time and showing transitions between species, supporting evolutionary theory. What is prezygotic isolation, and why is it important in speciation?
3, 2, 1, 0.
Prezygotic isolation prevents different species from mating, preserving species integrity and aiding in the formation of new species by reducing gene flow. Define frameshift mutation and describe its effects on protein synthesis.
3, 2, 1, 0.
A frameshift mutation is a genetic mutation caused by insertions or deletions of nucleotides that shift the reading frame, often resulting in non-functional proteins. What is an operon, and how does it function in prokaryotic gene regulation?
3, 2, 1, 0.
An operon is a cluster of genes regulated together by a single promoter and operator, allowing prokaryotes to control gene expression in response to environmental signals. Explain the Hardy-Weinberg principle and its assumptions.
3, 2, 1, 0.
The Hardy-Weinberg principle states that allele and genotype frequencies in a population remain constant if no evolution occurs. Assuming large population, no mutation, random mating, no gene flow, and no selection. What is the role of tRNA in the process of translation?
3, 2, 1, 0.
tRNA molecules transport specific amino acids to the ribosome, matching their anticodon with the mRNA codon to ensure correct amino acid incorporation in the growing polypeptide chain. How does Lamarck's theory of evolution differ from Darwin's?
3, 2, 1, 0.
Lamarck proposed that traits acquired during an organism's lifetime could be passed to offspring, while Darwin emphasized natural selection based on inherited traits. What is genetic counseling, and in what cases is it useful?
3, 2, 1, 0.
Genetic counseling provides information and support to individuals with genetic disorders or risks, guiding decisions on family planning and health management. Explain the role of restriction enzymes in genetic engineering.
3, 2, 1, 0.
Restriction enzymes cut DNA at specific sequences, enabling scientists to manipulate genes by cutting and pasting DNA segments for cloning or gene modification. What is homologous structure and its significance in evolution?
3, 2, 1, 0.
Homologous structures are body parts with similar anatomy but different functions in different species, indicating common ancestry and divergent evolution. Define genetic equilibrium and its implications for evolutionary stability.
3, 2, 1, 0.
Genetic equilibrium occurs when allele frequencies in a population remain stable, indicating no evolution and maintaining the current genetic makeup. What is genetic anticipation? And give an example.
3, 2, 1, 0.
Genetic anticipation refers to the phenomenon where symptoms of a genetic disorder become more severe or appear at an earlier age in successive generations, as seen in Huntington's disease. Describe the process of RNA splicing in eukaryotic cells.
3, 2, 1, 0.
RNA splicing removes introns from pre-mRNA, joining exons to form mature mRNA, allowing only the coding regions to be translated into proteins. What is gene silencing, and how can it affect gene expression?
3, 2, 1, 0.
Gene silencing is the suppression of gene expression through mechanisms like RNA interference, preventing translation of specific mRNA molecules. Define the concept of adaptive radiation with an example.
3, 2, 1, 0.
Adaptive radiation is the rapid diversification of a species into multiple forms to fill different ecological niches, as seen in Darwin's finches on the Galapagos Islands. How does antibiotic resistance develop in bacteria?
3, 2, 1, 0.
Antibiotic resistance arises when bacteria acquire mutations or genes that allow them to survive. Antibiotic exposure, often spread through horizontal gene transfer among bacteria. What are jumping genes or transposons, and who discovered them?
3, 2, 1, 0.
Jumping genes or transposons are DNA sequences that can change positions within the genome. Discovered by Barbara McClintock. Define analogous structures and provide an example.
3, 2, 1, 0.
Analogous structures have similar functions but different origins, like wings of birds and insects, and arise due to convergent evolution. Explain genetic recombination in bacteria.
3, 2, 1, 0.
Bacteria undergo genetic recombination through transformation, conjugation, and transduction, which increases genetic diversity without sexual reproduction. What is gene cloning and its significance?
3, 2, 1, 0.
Gene cloning is creating identical copies of a gene for research, medicine, or agriculture, allowing the study and manipulation of genes. How do multiple alleles differ from polygenic inheritance?
3, 2, 1, 0.
Multiple alleles involve more than two alternative forms of a gene for a trait, while polygenic inheritance involves multiple genes affecting a single trait, such as skin color. Define and give an example of pleiotropy.
3, 2, 1, 0.
Pleiotropy occurs when one gene influences multiple, seemingly unrelated phenotypic traits, as seen in the Marfan syndrome gene affecting both the heart and eyes. Describe the concept of genetic load.
3, 2, 1, 0.
Genetic load refers to the presence of unfavorable genetic mutations in a population, which can lower average fitness but may also allow for evolutionary flexibility. Explain how endosymbiosis contributed to the evolution of eukaryotes.
3, 2, 1, 0.
The endosymbiotic theory suggests that eukaryotic organelles like mitochondria and chloroplasts originated from symbiotic bacteria, leading to complex cells. Define epistasis and provide an example.
3, 2, 1, 0.
Epistasis is when one gene affects the expression of another gene, as seen in coat color in Labrador retrievers. where one gene can suppress another color gene. What is X inactivation and its role in genetic regulation?
3, 2, 1, 0.
X inactivation occurs in females to balance gene expression by randomly inactivating one of the two X chromosomes in each cell, leading to a mosaic expression of X-linked traits. Explain the significance of phylogenetic classification.
3, 2, 1, 0.
Phylogenetic classification groups organisms based on evolutionary ancestry and genetic relationships, helping scientists understand species origins and divergences. What is genetic erosion, and why is it a concern?
3, 2, 1, 0.
Genetic erosion is the loss of genetic diversity due to factors like habitat destruction or selective breeding, reducing resilience to environmental changes or diseases. Describe the effect of mutagens on DNA.
3, 2, 1, 0.
Mutagens are agents like chemicals or radiation that cause changes in DNA, potentially leading to genetic mutations, cancer, or inherited genetic diseases. What is gene flow and its role in evolution?
3, 2, 1, 0.
Gene flow is the transfer of alleles between populations through migration, which can introduce genetic diversity and reduce genetic differences between populations. Explain the role of chloroplast DNA in supporting endosymbiotic theory.
3, 2, 1, 0.
Chloroplast DNA resembles prokaryotic DNA in structure and gene sequences, supporting the idea that chloroplasts originated from symbiotic bacteria. Define conjugation and its role in bacterial evolution.
3, 2, 1, 0.
Conjugation is a process where bacteria transfer DNA through direct contact, allowing the spread of genes, including antibiotic resistance, within bacterial populations. What is a monohybrid cross, and what ratio does it yield in Mendelian inheritance?
3, 2, 1, 0.
A monohybrid cross examines one trait in a genetic cross, yielding a 3:1 phenotypic ratio in Mendel's first generation for a dominant recessive trait. Explain the role of ribosomes in protein synthesis.
3, 2, 1, 0.
Ribosomes facilitate the assembly of amino acids into proteins based on mRNA instructions, with tRNA bringing specific amino acids for each codon. What is a mutation hotspot, and why is it significant?
3, 2, 1, 0.
Mutation hotspots are DNA regions with high mutation rates, often leading to genetic disorders or increased variability in these areas, impacting evolution and disease susceptibility. Describe the inheritance pattern in hemophilia.
3, 2, 1, 0.
Hemophilia is an X-linked recessive disorder, primarily affecting males, with females as carriers. Affected males inherit the mutated gene from their mothers. Define polymorphism in genetics.
3, 2, 1, 0.
Polymorphism is the occurrence of multiple alleles or genetic forms within a population, maintaining genetic diversity and adaptability, as seen in human blood types. How does mitochondrial DNA support maternal inheritance?
3, 2, 1, 0.
Mitochondrial DNA is inherited only from the mother because sperm mitochondria are typically destroyed after fertilization, allowing tracking of maternal lineage. Explain the founder effect with an example.
3, 2, 1, 0.
The founder effect occurs when a small group forms a new population with limited genetic diversity, like the Amish, who have a higher frequency of certain genetic disorders. What is a karyotype, and how is it used in genetics?
3, 2, 1, 0.
A karyotype is an organized profile of an individual's chromosomes, used to detect chromosomal abnormalities and study evolutionary relationships between species. Define the term genetic bottleneck and its potential consequences.
3, 2, 1, 0.
A genetic bottleneck is a sharp reduction in population size, reducing genetic diversity, which can impact the population's ability to adapt to new environmental pressures. Describe the structure and role of telomeres.
3, 2, 1, 0.
Telomeres are protective caps at chromosome ends that prevent DNA loss during replication, playing a key role in cellular aging and stability. What is hybridization, and why is it significant in evolution?
3, 2, 1, 0.
Hybridization is the interbreeding of different species or varieties, producing hybrids that can introduce new genetic combinations and lead to speciation. How do fossils provide evidence of evolution?
3, 2, 1, 0.
Fossils show physical characteristics of ancient organisms and changes over time, revealing evolutionary transitions and common ancestry with modern species. Explain the concept of directional selection.
3, 2, 1, 0.
Directional selection favors one extreme phenotype, shifting the population's traits in one direction, as seen in peppered moths adapting to polluted environments. What are chromosomal aberrations, and give an example?
3, 2, 1, 0.
Chromosomal aberrations are structural changes in chromosomes, such as deletions, duplications, or translocations, as seen in conditions like Down syndrome. What is polyploidy, and how does it occur?
3, 2, 1, 0.
Polyploidy is a condition where organisms have multiple sets of chromosomes, resulting from errors in cell division, mitosis or meiosis, that double the chromosome number. Explain the difference between autosomes and sex chromosomes.
3, 2, 1, 0.
Autosomes are non-sex chromosomes present in pairs in both males and females, while sex chromosomes determine the sex of an individual, differing in males and females. XX and XY in humans. Describe how a dihybrid cross demonstrates independent assortment.
3, 2, 1,
A dihybrid cross between two heterozygous individuals for two traits demonstrates Mendel's law of independent assortment, showing a 9:3, 3:1 phenotypic ratio in the F2 generation. What is genetic drift, and how does it affect small populations?
3, 2, 1, 0.
Genetic drift is a random change in allele frequencies, having a more significant impact on small populations, potentially leading to the loss of genetic diversity. Define punctuated equilibrium in evolutionary theory.
3, 2, 1, 0.
Punctuated equilibrium suggests that evolution occurs in rapid bursts of change followed by long periods of stasis, as opposed to a gradual and continuous process. What is the role of the promoter region in gene transcription?
3, 2, 1, 0.
The promoter is a DNA sequence that initiates gene transcription by allowing RNA polymerase to bind and start RNA synthesis, crucial for gene regulation. Explain the concept of speciation.
3, 2, 1, 0.
Speciation is the process by which new species arise, often through geographic, reproductive, or ecological isolation, leading to divergence from a common ancestor. How does gene duplication contribute to evolution?
3, 2, 1, 0.
Gene duplication creates extra copies of genes, allowing one copy to accumulate mutations and potentially develop new functions without affecting the original gene's role. Define convergent evolution and give an example.
3, 2, 1, 0.
Convergent evolution occurs when unrelated species evolve similar traits due to similar environmental pressures, like the streamlined bodies of sharks and dolphins. What is genetic recombination, and why is it essential in meiosis?
3, 2, 1, 0.
Genetic recombination during meiosis involves the exchange of genetic material between homologous chromosomes, creating genetic diversity in offspring. Describe the role of DNA polymerase in DNA replication.
3, 2, 1, 0.
DNA polymerase synthesizes new DNA strands by adding nucleotides complementary to the template strand, ensuring accurate DNA replication and repair. Explain the term genetic code and its characteristics.
3, 2, 1, 0.
The genetic code is the set of rules by which information encoded in DNA is translated into proteins. It is universal, redundant, and specific, ensuring accurate protein synthesis. What is the role of the anticodon in protein synthesis?
3, 2, 1, 0.
The anticodon is a sequence of 3 nucleotides in tRNA that pairs with the complementary codon in mRNA, ensuring that the correct amino acid is added during protein synthesis. How does crossing over contribute to genetic variation?
3, 2, 1, 0.
Crossing over during prophase I of meiosis involves the exchange of genetic material between Homologous chromosomes. Increasing genetic diversity in gametes. What is the significance of Hardy-Weinberg equilibrium in population genetics?
3, 2, 1, 0.
Hardy-Weinberg equilibrium provides a model to study allele frequencies in a non-evolving population, helping to identify factors like selection and mutation that cause evolutionary change. Define sexual selection and provide an example.
3, 2, 1, 0.
Sexual selection is a type of natural selection where individuals with certain traits have an advantage in attracting mates, as seen in peacocks with bright feathers. Describe homologous chromosomes and their role in inheritance.
3, 2, 1, 0.
Homologous chromosomes are pairs of chromosomes with the same genes but possibly different alleles, one from each parent, which segregate independently during meiosis. What is a DNA mutation, and how can it impact an organism?
3, 2, 1, 0.
DNA mutations are changes in the genetic sequence that can be beneficial, neutral, or harmful, potentially leading to genetic disorders, increased adaptation, or no observable effect. Explain the concept of genetic mapping and its purpose.
3, 2, 1, 0.
Genetic mapping determines the position of genes on a chromosome, helping to identify gene linkages, predict inheritance patterns, and study gene function. How does natural selection drive evolution?
3, 2, 1, 0.
Natural selection favors individuals with advantageous traits, increasing their survival and reproduction rates, gradually changing allele frequencies in the population over generations. Define heterozygote advantage with an example.
3, 2, 1, 0.
Heterozygote advantage occurs when heterozygous individuals have greater fitness than homozygous individuals, as seen in sickle cell trait providing malaria resistance in heterozygotes. What is the significance of ribosomal RNA, rRNA, in evolution?
3, 2, 1, 0.
rRNA is highly conserved across species, making it useful for studying evolutionary relationships and tracing the ancestry of diverse life forms. Describe the role of a pedigree chart in genetic studies.
3, 2, 1, 0.
Pedigree charts trace the inheritance patterns of traits across generations, helping to identify genetic disorders and predict the probability of traits in offspring. What is genetic recombination, and why is it important?
3, 2, 1, 0.
Genetic recombination introduces new gene combinations during meiosis, increasing genetic diversity and helping populations adapt to changing environments. Explain linkage and how it affects inheritance patterns.
3, 2, 1, 0.
Linkage is when genes are located close together on a chromosome, causing them to be inherited together more often than not, reducing independent assortment. Define biodiversity in the context of genetics and evolution.
3, 2, 1, 0.
Biodiversity refers to the variety of life forms within a region, including genetic diversity within species, contributing to ecosystem resilience and adaptability. Describe a karyotype and its application in genetics.
3, 2, 1, 0.
A karyotype is an image of an individual's chromosomes arranged by size and shape, used to identify chromosomal abnormalities and study genetic disorders. What is RNA interference, NEE, and how does it regulate genes?
3, 2, 1, 0.
RNA interference, NEE, is a biological process where RNA molecules inhibit gene expression by degrading mRNA or blocking its translation, playing a role in gene regulation. Explain the founder effect and how it contributes to genetic drift.
3, 2, 1, 0.
The founder effect occurs when a small group establishes a new population with limited genetic variation, amplifying specific alleles due to genetic drift. How does codominance differ from incomplete dominance?
3, 2, 1, 0.
In codominance, both alleles are fully expressed, e.g., AB blood type, while in incomplete dominance, the phenotype is an intermediate blend of both alleles, e.g., pink flowers from red and white parents. What is the function of the Casparian strip in root endodermis?
3, 2, 1, 0.
The Casparian strip blocks the apoplastic flow of water and minerals, forcing them to enter the symplastic pathway for selective uptake. How does carbon dioxide concentration affect the rate of photosynthesis?
3, 2, 1, 0.
Increasing CO2 concentration boosts photosynthesis up to a certain point, after which other factors like light or temperature become limiting. What are the key components of the Z scheme in photosynthesis?
3, 2, 1, 0.
The Z scheme involves the flow of electrons from photosystem II to photosystem I, generating ATP and NADPH in the light-dependent reactions. How does abscisic acid, ABA, regulate stomatal closure?
3, 2, 1, 0.
Abscisic acid triggers the efflux of potassium ions from guard cells, leading to water loss and stomatal closure, especially under drought stress. How do legumes form symbiotic relationships with nitrogen-fixing bacteria?
3, 2, 1, 0.
Legumes form root nodules that house nitrogen-fixing rhizobium bacteria, which convert atmospheric nitrogen into ammonia, benefiting both the plant and bacteria. What is photolysis of water, and where does it occur?
3, 2, 1, 0.
Photolysis is the splitting of water molecules into oxygen, protons, and electrons using light energy, occurring in photosystem II. How does the pressure flow hypothesis explain translocation in the phloem?
3, 2, 1, 0.
The pressure flow hypothesis suggests that sugars are actively loaded into phloem at source tissues, creating a pressure gradient that drives the flow to sink tissues. What are the factors that affect the rate of transpiration?
3, 2,
The rate of transpiration is affected by factors like light intensity, temperature, humidity, wind speed, and the number of open stomata. What is the role of ATP in active transport across cell membranes?
3, 2, 1, 0.
ATP provides energy for the active transport of ions and molecules across cell membranes, moving them against their concentration gradient. How do the Calvin cycle and Hatch-Slack pathway differ in terms of their enzymes?
3, 2, 1, 0.
The Calvin cycle uses Rubisco for carbon fixation, while the Hatch-Slack C4 pathway uses PEP carboxylase to initially fix CO2 in mesophyll cells. What is the difference between photorespiration and normal respiration in plants?
3, 2, 1, 0.
Photorespiration occurs in the presence of light when Rubisco fixes oxygen instead of CO2, wasting energy, while normal respiration breaks down glucose to produce energy. What is apical dominance, and which hormone regulates it?
3, 2, 1, 0.
Apical dominance is the suppression of lateral bud growth by the shoot apex, primarily regulated by auxins produced at the tip. How does water potential influence osmosis in plant cells?
3, 2, 1, 0.
Water potential determines the direction of water movement. Water moves from regions of higher water potential to regions of lower water potential. How do plants maintain turgor pressure?
3, 2, 1, 0.
Plants maintain turgor pressure by absorbing water through osmosis into vacuoles, which press against the cell wall, keeping cells rigid and preventing wilting. What is the significance of cyclic and non-cyclic photophosphorylation in plants?
3, 2, 1, 0.
Non-cyclic photophosphorylation produces both ATP and NADPH, while cyclic photophosphorylation produces only ATP, ensuring a balance for the Calvin cycle. What is the role of cytokinins in cell division?
3, 2, 1, 0.
Cytokinins promote cell division by stimulating mitosis and are involved in the regulation of growth, development, and the delay of senescence in plants. How does vernalization influence flowering in plants?
3, 2, 1, 0.
Vernalization is the induction of flowering by prolonged exposure to cold temperatures, which triggers physiological changes necessary for flowering. How does the enzyme Rubisco function differently in the presence of oxygen?
3, 2, 1, 0.
In the presence of oxygen, Rubisco catalyzes the oxygenation of RUBP, leading to photorespiration, which reduces photosynthetic efficiency. How do aquaporins facilitate water movement in plants?
3, 2, 1, 0.
Aquaporins are channel proteins in cell membranes that allow the rapid movement of water molecules, facilitating osmosis and water balance. How does the Q10 coefficient explain the effect of temperature on metabolic processes in plants?
3, 2, 1, 0.
The Q10 coefficient measures the rate of metabolic reactions with a 10 degrees Celsius increase in temperature. Most metabolic processes in plants follow this relationship, with rates doubling or tripling as temperature rises. How does phloem loading occur during translocation?
3, 2, 1, 0.
Phloem loading occurs when sugars, mainly sucrose, are actively transported into sieve tube elements at the source, increasing the osmotic pressure and driving the flow towards sink tissues. What is etiolation, and how does it affect plant growth?
3, 2, 1, 0.
Etiolation is the growth of plants in the absence of light, characterized by elongated stems, small leaves, and pale color due to the lack of chlorophyll. What are the physiological effects of auxins on root development?
3, 2, 1, 0.
Auxins promote root initiation and elongation, and their concentration gradient affects the direction of root growth, often encouraging gravitropic responses. How does chlorophyll absorb light for photosynthesis?
3, 2, 1, 0.
Chlorophyll absorbs light most efficiently in the blue and red regions of the spectrum, using the energy for photosynthesis. What is the significance of water potential in plants?
3, 2, 1, 0.
Water potential determines the direction of water movement in plants, from regions of higher to lower water potential. How does the C4 pathway reduce photorespiration?
3, 2, 1, 0.
The C4 pathway spatially separates CO2 fixation and the Calvin cycle, concentrating CO2 in bundle sheath cells and minimizing oxygenation by rubisco. How does potassium regulate stomatal movement?
3, 2, 1, 0.
Potassium ions move into guard cells, causing them to take up water, swell, and open stomata. The reverse leads to stomatal closure. How do auxins influence phototropism in plants?
3, 2, 1, 0.
Auxins accumulate on the shaded side of a plant, promoting cell elongation and causing the plant to bend toward light. What is the role of symplastic and apoplastic pathways in mineral transport?
3, 2, 1, 0.
The symplastic pathway moves minerals through the cytoplasm of plant cells via plasmodesmata, while the apoplastic pathway moves them through cell walls and intercellular spaces. How does the Calvin cycle use ATP and NADPH?
3, 2, 1, 0.
ATP and NADPH produced in the light-dependent reactions are used in the Calvin cycle to fix CO2 and produce glucose. What is the function of PEP carboxylase in C4 and CAM plants?
3, 2, 1, 0.
PEP carboxylase fixes CO2 into a 4-carbon compound, preventing photorespiration and ensuring efficient carbon fixation in C4 and CAM plants. How does transpiration affect the movement of water in plants?
3, 2, 1, 0.
Transpiration creates a negative pressure in the leaves, pulling water upward through the xylem from the roots via cohesion and adhesion. How do brasinosteroids influence plant growth?
3, 2, 1, 0.
Brasinosteroids promote cell elongation, division, and differentiation, and they also help in stress responses in plants. What is the role of cyclic electron transport in photosynthesis?
3, 2, 1, 0.
Cyclic electron transport generates ATP without producing NADPH by cycling electrons through photosystem I and back to the electron transport chain. How do sieve tube elements in the phloem function during translocation?
3, 2, 1, 0.
Sieve tube elements transport sugars and other organic nutrients from source tissues, like leaves, to sink tissues, like roots. How do plants adapt to water stress using abscisic acid?
3, 2, 1, 0.
Abscisic acid promotes stomatal closure and induces dormancy in seeds to help plants conserve water during drought. What is the importance of respiration in plants?
3, 2, 1, 0.
Respiration in plants breaks down carbohydrates into CO2 and water, releasing energy for growth and other metabolic activities. How do guard cells control the opening and closing of stomata?
3, 2, 1, 0.
Guard cells change shape due to the osmotic flow of water, driven by ion movement, to regulate the opening and closing of stomata. What is the function of NADP+ in photosynthesis?
3, 2, 1, 0.
NADP+ accepts electrons during the light-dependent reactions to form NADPH, which is used in the Calvin cycle for CO2 fixation. How do auxins and cytokinins interact in plant growth?
3, 2, 1, 0.
Auxins promote cell elongation and root formation, while cytokinins promote cell division and shoot development. Their balance regulates organogenesis. What role do endodermal cells play in selective absorption in plants?
3, 2, 1, 0.
Endodermal cells, with their Casparian strips, regulate the selective absorption of water and minerals into the xylem. How does sucrose loading into the phloem affect translocation?
3, 2, 1, 0.
Sucrose loading into the phloem increases osmotic pressure, causing water to enter and generate a pressure flow that moves the sugar solution to sink tissues. How does oxygen concentration affect photorespiration?
3, 2, 1, 0.
Higher oxygen concentration increases the likelihood of rubisco binding with O2 instead of CO2, leading to photorespiration, which wastes energy. What is the significance of crassylation acid metabolism, CAM, in plants?
3, 2, 1, 0.
CAM is a photosynthetic adaptation in some plants that allows them to fix CO2 at night, reducing water loss in arid environments. How do temperature extremes affect enzyme activity in plants?
3, 2, 1, 0.
Extreme temperatures can denature enzymes, reducing their activity. while optimal temperatures enhance their catalytic functions. What is the function of cytochromes in the electron transport chain of plant cells?
3, 2, 1, 0.
Cytochromes are proteins in the electron transport chain that transfer electrons and help pump protons across the membrane to generate ATP. How do hydrophytes adapt to aquatic environments?
3, 2, 1, 0.
Hydrophytes, or water plants, have adaptations such as thin cuticles, large air spaces for buoyancy, and reduced vascular tissues. What is the role of carotenoids in photosynthesis?
3, 2, 1, 0.
Carotenoids absorb light in wavelengths that chlorophyll cannot, extending the range of light that can drive photosynthesis. How does the leaf anatomy of xerophytes help them conserve water?
3, 2, 1, 0.
Xerophytes have thick cuticles, sunken stomata, and reduced leaf surface area to minimize water loss in arid conditions. What is the role of phototropins in plant movement?
3, 2, 1, 0.
Phototropins are light receptors that detect blue light, triggering phototropic bending of plants toward the light source. How does the light saturation point affect the rate of photosynthesis?
3, 2, 1, 0.
The light saturation point is the intensity at which photosynthesis reaches its maximum rate. Beyond this point, light no longer increases the rate. What is the role of intercellular air spaces in plant leaves?
3, 2, 1, 0.
Intercellular air spaces in leaves allow for the diffusion of gases, CO2 and O2, during photosynthesis and respiration. How does aerobic respiration differ from anaerobic respiration in plants?
3, 2, 1, 0.
Aerobic respiration requires oxygen and produces more ATP by fully oxidizing glucose, while anaerobic respiration occurs without oxygen, producing less ATP and yielding byproducts like ethanol. How does gibberellin affect stem elongation?
3, 2, 1, 0.
Gibberellins promote stem elongation by stimulating cell division and elongation in the internodes. How does leaf abscission occur, and what hormones are involved?
3, 2, 1, 0.
Leaf abscission occurs when auxin levels decrease and ethylene increases, leading to the breakdown of cells in the abscission zone. What is the role of light in photoperiodism?
3, 2, 1, 0.
Photoperiodism is the response of plants to the length of day and night, affecting flowering and other developmental processes. How do plants exhibit thymotropism?
3, 2, 1, 0.
Thymotropism is the growth response to touch, seen in climbing plants like tendrils that wrap around objects for support. How does the Kranz anatomy of C4 plants reduce photorespiration?
3, 2, 1, 0.
Kranz anatomy in C4 plants has bundle sheath cells surrounding the vascular bundles, where CO2 is concentrated. reducing photorespiration. How does the enzyme pyruvate dehydrogenase contribute to cellular respiration?
3, 2, 1, 0.
Pyruvate dehydrogenase converts pyruvate into acetyl-CoA, which enters the Krebs cycle during aerobic respiration. How do leguminous plants enrich soil fertility?
3, 2, 1, 0.
Leguminous plants form symbiotic associations with nitrogen-fixing bacteria, converting atmospheric nitrogen into ammonia, which enriches soil. What is the difference between macronutrients and micronutrients in plants?
3, 2, 1, 0.
Macronutrients are needed in large quantities, e.g., nitrogen, potassium, while micronutrients are required in smaller amounts, e.g., iron, zinc. How does temperature affect the rate of photosynthesis?
3, 2, 1, 0.
The rate of photosynthesis increases with temperature up to an optimal point, after which enzymes denature, and the rate decreases. How does the cohesion tension theory explain water transport in plants?
3, 2, 1, 0.
The cohesion tension theory states that water is pulled up the plant through the xylem by the cohesive properties of water molecules and the tension created by transpiration. What is the role of ferritin in plants?
3, 2, 1, 0.
Ferritin stores iron in plants and releases it when needed for essential functions like chlorophyll synthesis. How does the enzyme nitrogenase function in nitrogen fixation?
3, 2, 1, 0.
Nitrogenase is the enzyme responsible for converting atmospheric nitrogen, N2, into ammonia, NH3, in nitrogen-fixing bacteria. How do CAM plants differ from C4 plants in their adaptation to arid conditions?
3, 2, 1, 0.
CAM plants fix CO2 at night and store it as malic acid, while C4 plants spatially separate CO2 fixation and the Calvin cycle. What role do gibberellins play in seed germination?
3, 2, 1, 0.
Gibberellins stimulate the production of enzymes that break down starch into sugars, providing energy for seed germination. What is the difference between water potential and osmotic potential?
3, 2, 1, 0.
Water potential is the potential energy of water in a system, while osmotic potential is the component of water potential due to solute concentration. How does temperature affect enzyme activity in plant metabolism?
3, 2, 1, 0.
Enzyme activity increases with temperature up to an optimum level. Beyond this, high temperatures denature the enzymes, reducing their activity. How does auxin promote apical dominance in plants?
3, 2, 1, 0.
Auxin produced at the shoot apex inhibits the growth of lateral buds, maintaining apical dominance and directing vertical growth. What is the significance of turgor pressure in plants?
3, 2, 1, 0.
Turgor pressure maintains the rigidity of plant cells, providing structural support and aiding in cell expansion and growth. How do mycorrhizal associations benefit plants?
3, 2, 1, 0.
Mycorrhizal fungi enhance nutrient and water absorption in plants by extending the surface area of their roots. What is the role of cytokinin in delaying leaf senescence?
3, 2, 1, 0.
Cytokinins delay leaf senescence by promoting nutrient mobilization and maintaining chlorophyll levels in leaves. How do guard cells regulate transpiration in plants?
3, 2, 1, 0.
Guard cells control the opening and closing of stomata, thus regulating water loss through transpiration based on environmental conditions. How does the C3 pathway differ from the C4 pathway in photosynthesis?
3, 2, 1, 0.
The C3 pathway directly fixes CO2 using Rubisco in the Calvin cycle, while the C4 pathway initially fixes CO2 into a 4-carbon compound before the Calvin cycle. How do plants absorb nutrients from the soil?
3, 2, 1, 0.
Nutrients are absorbed through root hairs via active and passive transport processes, including ion exchange and diffusion. What is the significance of respiratory quotient, RQ, in plant respiration?
3, 2, 1, 0.
The respiratory quotient, RQ, is the ratio of CO2 produced to O2 consumed during respiration, indicating the type of substrate being metabolized. How do xerophytes adapt to arid environments?
3, 2, 1, 0.
Xerophytes have adaptations like thick cuticles, sunken stomata, and reduced leaf surface area to minimize water loss. How do auxins influence cell elongation?
3, 2, 1, 0.
Auxins promote cell elongation by increasing the plasticity of the cell wall, allowing it to stretch during growth. How do plants use ATP during active transport?
3, 2, 1, 0.
ATP provides energy to pump ions and molecules across cell membranes against their concentration gradient, enabling active transport. How do temperature and humidity affect transpiration rates in plants?
3, 2, 1, 0.
High temperatures increase transpiration rates by causing more evaporation, while high humidity decreases transpiration due to reduced water vapor gradient. What is the role of plastocyanin in the electron transport chain?
3, 2, 1, 0.
Plastocyanin transfers electrons between cytochrome B6F complex and photosystem I during the light-dependent reactions of photosynthesis. How do plants regulate water loss through stomatal movement?
3, 2, 1, 0.
Stomatal opening and closing are controlled by changes in turgor pressure in guard cells, influenced by factors like light, CO2 levels, and water availability. What is the significance of proton pumps in plant cells?
3, 2, 1, 0.
Proton pumps create an electrochemical gradient by pumping H ions out of the cell, which drives the active transport of nutrients and ions. How does photorespiration affect the efficiency of photosynthesis?
3, 2, 1, 0.
Photorespiration decreases the efficiency of photosynthesis by diverting energy and reducing carbon fixation when Rubisco reacts with oxygen instead of CO2. What is the function of Rubisco in photosynthesis?
3, 2, 1, 0.
Rubisco catalyzes the fixation of CO2 in the Calvin cycle, producing a 3-carbon compound, PGA, in the C3 pathway. What are the components of the phloem tissue in plants?
3, 2, 1, 0.
Phloem tissue consists of sieve tube elements, companion cells, phloem fibers, and phloem parenchyma, which are involved in transporting nutrients. How do enzymes in the Calvin cycle regenerate RUBP? In the Calvin cycle, RUBP, ribulose bisphosphate, is regenerated using ATP, allowing the cycle to continue fixing CO2. What is the role of the Casparian strip in the root?
3, 2, 1, 0.
The Casparian strip in the root endodermis blocks the apoplastic pathway, forcing water and nutrients to pass through the selectively permeable plasma membrane. How do aquaporins facilitate water transport in plant cells?
3, 2, 1, 0.
Aquaporins are membrane proteins that form channels for the passive transport of water across cell membranes. How do proton gradients drive ATP synthesis in chloroplasts?
3, 2, 1, 0.
Proton gradients created across the thylakoid membrane during the light-dependent reactions drive ATP synthesis by ATP synthase. What is the role of peroxisomes in photorespiration?
3, 2, 1, 0.
Peroxisomes metabolize glycolate produced during photorespiration, converting it into glycine and later into serine. How do plants respond to drought stress at the cellular level?
3, 2, 1, 0.
Under drought stress, plants accumulate solutes like proline and close stomata to reduce water loss while protecting cellular structures. How does photoinhibition affect photosynthesis?
3, 2, 1, 0.
Photoinhibition occurs when excessive light damages the photosynthetic machinery, particularly photosystem II, reducing the rate of photosynthesis. What is the role of manganese in photosynthesis?
3, 2, 1, 0.
Manganese is a cofactor in the water-splitting reaction during the light-dependent reactions, where it helps release oxygen from water. How do guard cells maintain ion balance during stomatal movement?
3, 2, 1, 0.
Guard cells actively transport potassium ions to control their turgor pressure, leading to the opening or closing of stomata. How does the Calvin cycle differ between C3 and CAM plants?
3, 2, 1, 0.
In C3 plants, the Calvin cycle occurs during the day, while in CAM plants, CO2 is fixed at night, stored as malic acid. and used during the day for photosynthesis. How do plants regulate the uptake of mineral ions from the soil?
3, 2, 1, 0.
Plants regulate mineral uptake through active transport mechanisms involving specific ion channels and proton pumps in root cells. What is the function of boron in plant growth?
3, 2, 1, 0.
Boron is essential for cell wall formation, membrane integrity, and the regulation of carbohydrate transport in plants. How do leg hemoglobin and nitrogenase function in nitrogen fixation?
3, 2, 1, 0.
Leg hemoglobin binds oxygen, protecting nitrogenase, which catalyzes the conversion of atmospheric nitrogen, N2, into ammonia, NH3, in nitrogen-fixing nodules. How does the chloroplast structure facilitate photosynthesis?
3, 2, 1, 0.
Chloroplasts have a double membrane with internal thylakoids where light-dependent reactions occur, and the stroma where the Calvin cycle takes place. How do changes in light intensity affect stomatal movement?
3, 2, 1, 0.
Stomata open in response to light, particularly blue light, as it triggers potassium ion influx into guard cells, increasing their turgor pressure. What is the role of phosphorus in plant growth?
3, 2, 1, 0.
Phosphorus is a key component of ATP, nucleic acids, and phospholipids, playing a vital role in energy transfer and metabolic reactions. How do plants adapt to nutrient-deficient soils?
3, 2, 1, 0.
Plants adapt to nutrient-deficient soils through mechanisms such as forming symbiotic relationships with mycorrhiza, root nodules, or increasing root surface area. What is the role of ATP and NADPH in the Calvin cycle?
3, 2, 1, 0.
ATP and NADPH produced in the light-dependent reactions provide energy and reducing power for the fixation of CO2 and synthesis of carbohydrates in the Calvin cycle. What is the role of potassium in plant nutrition?
3, 2, 1, 0.
Potassium regulates osmosis, stomatal movement, enzyme activation, and the transport of sugars within plants. How do gibberellins promote seed germination?
3, 2, 1, 0.
Gibberellins trigger the production of enzymes like amylase, which breaks down stored starch into sugars to provide energy for germination. What is the significance of turgor pressure in guard cells for stomatal opening?
3, 2, 1, 0.
Turgor pressure in guard cells increases due to water influx when potassium ions accumulate, causing stomata to open. How does temperature impact seed dormancy and germination?
3, 2, 1, 0.
High temperatures can break seed dormancy by disrupting inhibitors like abscisic acid, while optimum temperatures promote germination by activating enzymes. How do water and nutrient uptake differ between monocots and dicots?
3, 2, 1, 0.
Monocots typically have fibrous root systems that spread out for shallow water and nutrient absorption, while dicots have taproots that allow for deeper absorption. What role does cytokinin play in cell division and differentiation?
3, 2, 1, 0.
Cytokinins promote cell division and differentiation by stimulating the synthesis of proteins and regulating the cell cycle. How does the electron transport chain function during oxidative phosphorylation in plant mitochondria?
3, 2, 1, 0.
The electron transport chain in mitochondria transfers electrons, pumping protons to generate a proton gradient that drives ATP synthesis. How does magnesium deficiency affect photosynthesis in plants?
3, 2, 1, 0.
Magnesium is a central component of chlorophyll. Its deficiency results in chlorosis and impaired photosynthesis. What is the role of ATP synthase in the chloroplast?
3, 2, 1, 0.
ATP synthase in the chloroplast generates ATP during the light-dependent reactions by utilizing the proton gradient created across the thylakoid membrane. How does pH affect enzyme activity in photosynthesis and respiration?
3, 2, 1, 0.
Enzymes in photosynthesis and respiration have an optimal pH range. Outside this range, enzyme activity declines, affecting metabolic processes. How do plants respond to waterlogging stress?
3, 2, 1, 0.
In response to waterlogging, some plants develop parenchyma tissue, which helps transport oxygen to submerged roots. What is the role of transpirational pull in water transport?
3, 2, 1, 0.
Transpirational pull creates a negative pressure in the leaves, drawing water upward from the roots through the xylem. How does calcium influence cell wall formation in plants?
3, 2, 1, 0.
Calcium helps in the formation of calcium pectate, which is essential for maintaining the structural integrity of plant cell walls. What is the function of plastoquinone in the photosynthetic electron transport chain?
3, 2, 1, 0.
Plastoquinone transfers electrons from photosystem II to the cytochrome B6F complex in the electron transport chain. How do plants regulate the balance between photosynthesis and respiration?
3, 2, 1, 0.
Plants balance photosynthesis and respiration by modulating enzyme activity and stomatal opening, based on light, temperature, and internal energy needs. What is the role of phytochromes in plant growth?
3, 2, 1, 0.
Phytochromes are light receptors that regulate processes like seed germination, stem elongation, and flowering based on light conditions. How does phototropism help plants?
3, 2, 1, 0.
Phototropism allows plants to grow toward light sources, optimizing photosynthesis by positioning leaves for maximum light absorption. How do plants maintain osmotic balance during drought conditions?
3, 2, 1, 0.
Plants accumulate osmolytes like proline and sugars to maintain osmotic balance, preventing water loss during drought. How do sink and source relations affect phloem transport?
3, 2, 1, 0.
Phloem transport depends on the source. Leaves, where sugars are produced, and sink, roots, fruits, where sugars are used or stored. What is the role of ATP in active ion uptake in plants?
3, 2, 1, 0.
ATP provides the energy for active transport of ions against their concentration gradients, especially in root cells. How do cytokinins affect apical dominance?
3, 2, 1, 0.
Cytokinins counteract the inhibitory effect of auxins on lateral bud growth, reducing apical dominance and promoting branching. How does the cuticle reduce water loss in plants?
3, 2, 1, 0.
The waxy cuticle forms a barrier that reduces water loss through the epidermis by limiting evaporation. What is the role of nitrate reductase in nitrogen assimilation?
3, 2, 1, 0.
Nitrate reductase catalyzes the reduction of nitrate, number 3, to nitrite, number 2, during nitrogen assimilation in plants. How do plants use ethylene to respond to mechanical stress?
3, 2, 1, 0.
Ethylene promotes stem thickening and reduced elongation in response to mechanical stress, like wind or touch, helping plants withstand stress. How do photosystem I and photosystem II differ in function?
3, 2, 1, 0.
Photosystem II absorbs light and splits water molecules to release oxygen, while photosystem I absorbs light to produce NADPH in photosynthesis. What is the role of potassium ions in opening stomata?
3, 2, 1, 0.
Potassium ions accumulate in guard cells, increasing their osmotic pressure, leading to water influx and stomatal opening. How does abscisic acid help plants survive drought conditions?
3, 2, 1, 0.
Abscisic acid induces stomatal closure to reduce water loss and triggers dormancy in seeds during drought conditions. How does oxygen inhibit nitrogenase activity in nitrogen fixation?
3, 2, 1, 0.
Oxygen inhibits nitrogenase activity by binding to it. Thus leguminous plants maintain low oxygen levels in root nodules with leg hemoglobin. How does the enzyme RuBisCO exhibit dual activity in plants?
3, 2, 1, 0.
RuBisCO-ACTS is both a carboxylase, fixing CO2, and an oxygenase, fixing O2 during photorespiration, affecting photosynthetic efficiency. What is the significance of chloroplast stroma in the Calvin cycle?
3, 2, 1, 0.
The stroma contains enzymes for the Calvin cycle, where CO2 is fixed into organic compounds like glucose. How does stomata respond to light and CO2 concentration?
3, 2, 1, 0.
Stomata open in response to light. especially blue light, and low internal CO2 concentration, allowing gas exchange. How do C3 plants differ from CAM plants in terms of CO2 fixation?
3, 2, 1, 0.
C3 plants fix CO2 during the day through the Calvin cycle, while CAM plants fix CO2 at night and store it as malate to use during the day. How does water potential influence water movement in plants?
3, 2, 1, 0.
Water moves from regions of higher water potential to lower water potential, driving water transport in the plant. How does zinc deficiency affect plant growth?
3, 2, 1, 0.
Zinc is required for the synthesis of auxins and certain enzymes. Its deficiency leads to stunted growth and reduced internode length. How do plants excrete waste products?
3, 2, 1, 0.
Plants excrete waste products through diffusion, storage in vacuoles, shedding leaves, or exudation of resins and gums. What is the function of cytochrome B6F complex in photosynthesis?
3, 2, 1, 0.
The cytochrome B6F complex transfers electrons between photosystem II and photosystem I, contributing to the proton gradient across the thylakoid membrane. How do gibberellins influence fruit development?
3, 2, 1, 0.
Gibberellins promote fruit growth and development by enhancing cell elongation and division in fruits. How does passive transport differ from active transport in plants?
3, 2, 1, 0.
Passive transport involves the movement of molecules along their concentration gradient without energy input, while active transport requires ATP to move molecules against their gradient. What is the role of mitochondrial cristae in respiration?
3, 2, 1, 0.
The cristae increase the surface area for the electron transport chain, where ATP is produced during oxidative phosphorylation. How do halophytes adapt to saline environments?
3, 2, 1, 0.
Halophytes secrete excess salts through specialized salt glands and accumulate compatible solutes, like proline, to maintain osmotic balance. What is the function of NADP in photosynthesis?
3, 2, 1, 0.
NADP is the final electron acceptor in the light reactions, becoming NADPH, which is used in the Calvin cycle for carbon fixation. How does transpiration help in nutrient transport in plants?
3, 2, 1, 0.
Transpiration creates a negative pressure in the xylem, helping to pull water and dissolved nutrients from the roots to the leaves. How do plants store the products of photosynthesis?
3, 2, 1, 0.
Plants store the products of photosynthesis, such as glucose, in the form of starch in leaves, stems, roots, and seeds. What is the function of ATP during glycolysis in plant cells?
3, 2, 1, 0.
ATP provides the energy to phosphorylate glucose during the first steps of glycolysis, allowing the breakdown of glucose into pyruvate. How does the Kranz anatomy of C4 plants improve their efficiency? The Kranz anatomy allows spatial separation of CO2 fixation and the Calvin cycle, reducing photorespiration in C4 plants. What is the role of endodermis in selective nutrient uptake?
3, 2, 1, 0.
The endodermis, with its Casparian strip, controls the selective uptake of minerals by forcing water to pass through the plasma membrane. How does photoperiodism regulate flowering in plants?
3, 2, 1, 0.
Photoperiodism involves the response of plants to the relative lengths of day and night, influencing the timing of flowering. How do antiport and symport differ in ion transport in plants?
3, 2, 1, 0.
Antiport moves two substances in opposite directions across a membrane, while symport moves them in the same direction. What role does iron play in plant metabolism? Iron is essential for chlorophyll synthesis and is a component of enzymes involved in respiration and nitrogen metabolism. How does succinate dehydrogenase function in the Krebs cycle?
3, 2, 1, 0.
Succinate dehydrogenase catalyzes the oxidation of succinate to fumarate in the Krebs cycle, generating FADH2. What is the role of xylem parenchyma in plants?
3, 2, 1, 0.
Xylem parenchyma stores nutrients and water, and it also aids in lateral conduction of water in plants. How does cyclic photophosphorylation differ from non-cyclic photophosphorylation? In cyclic photophosphorylation, only photosystem I is involved, and only ATP is produced, whereas non-cyclic involves both photosystems and produces ATP, NADPH, and oxygen. How do adventitious roots differ from taproots in function and structure? Adventitious roots arise from non-root tissues and provide additional support, while taproots grow from the radicle and anchor the plant deeply. How does the Q10 coefficient relate to plant respiration rates?
3, 2, 1, 0.
The Q10 coefficient describes the rate of respiration, which typically doubles with a 10 degrees Celsius rise in temperature. What is the role of auxins in apical dominance?
3, 2, 1, 0.
Auxins produced at the shoot apex suppress the growth of lateral buds, maintaining apical dominance. How does transpiration aid in nutrient distribution in plants?
3, 2, 1, 0.
Transpiration pulls water and dissolved nutrients upward from the roots to the leaves via the xylem. What is the role of the PEP carboxylase enzyme in C4 plants?
3, 2, 1, 0.
PEP carboxylase fixes CO2 into oxaloacetate in the mesophyll cells of C4 plants, which is later transferred to bundle sheath cells for the Calvin cycle. How does stomata respond to abscisic acid during water stress?
3, 2, 1,
Abscisic acid signals stomatal closure during water stress to prevent excessive water loss. How does sulfur deficiency manifest in plants?
3, 2, 1, 0.
Sulfur deficiency leads to chlorosis in younger leaves, stunted growth, and reduced protein synthesis in plants. What role does ATP play in the Calvin cycle?
3, 2, 1, 0.
ATP provides the energy for carbon fixation and the regeneration of RUBP in the Calvin cycle during photosynthesis. What is the role of proton pumps in root cells?
3, 2, 1, 0.
Proton pumps create an electrochemical gradient by actively pumping protons out of root cells, facilitating nutrient uptake via ion channels. How does auxin promote root initiation in plant cuttings?
3, 2, 1, 0.
Auxin promotes cell division and elongation, encouraging root formation in plant cuttings. How does leaf anatomy affect the rate of transpiration?
3, 2, 1, 0.
Leaves with thicker cuticles, fewer stomata, or sunken stomata reduce transpiration, while thinner cuticles increase it. What is the role of ethylene in fruit ripening?
3, 2, 1, 0.
Ethylene promotes the breakdown of cell walls, conversion of starches to sugars, and the degradation of chlorophyll, leading to fruit ripening. How does magnesium deficiency affect plant metabolism?
3, 2, 1, 0.
Magnesium is a central atom in chlorophyll. Its deficiency leads to reduced chlorophyll production and impaired photosynthesis. What is the role of the bundle sheath cells in C4 plants?
3, 2, 1, 0.
Bundle sheath cells in C4 plants are the site of the Calvin cycle, where CO2 released from malate is fixed into carbohydrates. How does the light saturation point affect photosynthesis?
3, 2, 1, 0.
The light saturation point is the intensity beyond which light no longer increases the rate of photosynthesis, as other factors become limiting. How does manganese contribute to photosynthetic reactions?
3, 2, 1, 0.
Manganese is a cofactor in the water-splitting complex of photosystem II, aiding in the release of oxygen from water. What is the significance of malate in cam plants?
3, 2, 1, 0.
Malate ACTS is a temporary storage form of CO2, fixed at night and used during the day for photosynthesis in cam plants. How does diffusion differ from osmosis in plant cells?
3, 2, 1, 0.
Diffusion is the movement of molecules from high to low concentration, while osmosis specifically refers to the movement of water across a semi-permeable membrane. What is the role of the enzyme amylase in seed germination?
3, 2, 1, 0.
Amylase breaks down stored starch in seeds into sugars, providing energy for seed germination. How do sieve tube elements facilitate transport in plants?
3, 2, 1, 0.
Sieve tube elements connected by sieve plates, allow the movement of organic nutrients like sugars through the phloem. How does temperature affect enzyme activity in photosynthesis?
3, 2, 1, 0.
Enzyme activity in photosynthesis typically increases with temperature up to an optimum point, after which it declines. How does copper deficiency affect plants?
3, 2, 1, 0.
Copper is essential for enzyme activity and chlorophyll synthesis. Its deficiency leads to stunted growth and wilting. How do xylem vessels differ from tracheids in structure and function?
3, 2, 1, 0.
Xylem vessels are larger and more efficient at water transport, while tracheids are narrower and provide structural support and water conduction. How do light reactions and dark reactions differ in photosynthesis?
3, 2, 1, 0.
Light reactions convert light energy into chemical energy, ATP and NADPH, while dark reactions, Calvin cycle, fix CO2 into organic compounds like glucose. How does phloem loading occur in plants?
3, 2, 1, 0.
Phloem loading involves the active transport of sucrose into sieve tubes from mesophyll cells, through symplast or apoplast pathways. What is the significance of turgor pressure in plant cells?
3, 2, 1, 0.
Turgor pressure provides structural support to plant cells, maintaining rigidity in tissues and aiding in stomatal opening. How does auxin affect cell elongation in shoots?
3, 2, 1, 0.
Auxin stimulates cell elongation by increasing the plasticity of cell walls, allowing cells to expand. How does nitrate assimilation occur in plants?
3, 2, 1, 0.
Nitrate is reduced to nitrite by nitrate reductase, then further reduced to ammonia by nitrite reductase for incorporation into amino acids. How do CAM plants minimize water loss during photosynthesis?
3, 2, 1, 0.
CAM plants open their stomata at night to fix CO2, storing it as malate, and close their stomata during the day to minimize water loss. What is the role of phytochrome in seed germination?
3, 2, 1, 0.
Phytochrome senses red and far red light, regulating seed germination based on light conditions. How does the proton gradient drive ATP synthesis in chloroplasts?
3, 2, 1, 0.
The proton gradient generated during electron transport in the thylakoid membrane drives ATP synthesis via ATP synthase. How do guard cells regulate stomatal opening and closing?
3, 2, 1, 0.
Guard cells swell with water, opening stomata, and lose water to close stomata, regulated by light, CO2 levels, and hormones. What is the role of calcium ions in plant signaling?
3, 2, 1, 0.
Calcium ions act as secondary messengers in various plant signaling pathways, including responses to environmental stimuli and hormones. How does the glyoxylate cycle support seed germination in fat-storing seeds?
3, 2, 1, 0.
The glyoxylate cycle converts stored fats into sugars, providing energy for seedling growth before photosynthesis begins. How does potassium affect enzyme activation in plant cells?
3, 2, 1, 0.
Potassium activates various enzymes involved in protein synthesis, photosynthesis, and respiration in plant cells. What is the function of the Casparian strip in roots?
3, 2, 1, 0.
The Casparian strip forces water and solutes to pass through the plasma membranes of endodermal cells, regulating the uptake of nutrients. What is the role of cytokinin in delaying senescence?
3, 2, 1, 0.
Cytokinins delay leaf senescence by promoting cell division and chloroplast activity, thus maintaining leaf functions. How does sulfur contribute to amino acid synthesis in plants?
3, 2, 1, 0.
Sulfur is a component of certain amino acids like cysteine and methionine, crucial for protein synthesis in plants. How do aquaporins facilitate water movement in plant cells?
3, 2, 1, 0.
Aquaporins are membrane proteins that facilitate the passive transport of water molecules across cell membranes. How does photorespiration affect photosynthetic efficiency in C3 plants?
3, 2, 1, 0.
Photorespiration consumes oxygen and releases CO2 without producing ATP or sugars, reducing the efficiency of photosynthesis. What role do sieve plates play in the phloem?
3, 2, 1, 0.
Sieve plates are perforated cell walls that allow the flow of phloem sap between sieve tube elements. How does temperature influence the rate of transpiration in plants?
3, 2, 1, 0.
Higher temperatures increase the rate of transpiration by increasing water vapor pressure and stomatal opening. How does photolysis of water contribute to the light reactions of photosynthesis?
3, 2, 1, 0.
Photolysis splits water molecules into oxygen, protons, and electrons, replenishing electrons lost by photosystem II during the light reactions. How does boron deficiency affect reproductive development in plants?
3, 2, 1, 0.
Boron deficiency impairs pollen tube growth, leading to poor fertilization and reduced seed and fruit development. How do reactive oxygen species, ROS, form during photosynthesis? And how do plants mitigate their effects?
3, 2, 1, 0.
ROS form as byproducts of electron transport. Plants mitigate ROS damage through antioxidants like superoxide dismutase. How does the Calvin cycle regenerate RUBP?
3, 2, 1, 0.
The Calvin cycle regenerates RUBP from glyceraldehyde-3-phosphate G3P through a series of reactions requiring ATP.